بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. Thank you everyone who attending this seminar. It's our IBD webinars on the biotechnology, science, business, and society webinars. And this webinar is 29th. Alhamdulillah, today we have a very special guest. Dr. Hussein bin Muhammad is the head of toxicology and pharmacology of the unit of the Herbal Medicine Research Center in IMR, which is in Selangor, Malaysia. His research interest is mainly for the in vivo safety study, which is very important, and I think it's everyone like nowadays for about the safety assessment of the herbal products or natural products. And for this very important research, we find this in Malaysia, this special laboratory, they are accredited from OECD as a good laboratory practice under the United Nations OECD and for non-clinical research facility. He served as a study as directors of contract research in general, toxicology, as well as managing of the clinical testing unit. And he have wide range experience and who is also expert panel of the Ministry of Science and Technology, mostly Malaysia. He is also as a particular cluster, this is also a panel member under the cluster of the National Key Economic Area under the Ministry of Agriculture and Food Industry. With his experience for many years, regulatory experience for releasing bioactive ingredients and approval, we are very happy today for having him in IBD webinars. Thank you very much, Dr. Hussein, again, for accepting our invitation.
Thank you so much, Prof. Hisham, for the introduction. So I feel so honored for me to be here to share with all of you my experience or the information regarding the toxicology studies, especially in herbal research. For today, basically, I'm not going to go into details, and I will share with all of you some of the information about the toxicology studies, about what of the toxicology studies needed, type or the study design, how to conduct the toxicology studies. And also I'm going to share some of the work that have been conducted in herbal research. Okay, before I start with my presentation, so as you know that we are not Institute for Medical Research. We are now moved to a new location situated in City Alam, Selangor, with the new building. As you know that before this, we were in Jalan Pahang with the old building. So now we move into the better place with the better facilities to do our research. Okay, so I'm from the Herbal Medicine Research Center. So basically our center, we consist of 5 different units. And with the function, the objective of our center is to provide the scientific evidence of the safety and efficacy of medicinal plant for development of herbal products. So, and currently our research activities include the preclinical and the clinical evaluation of the herbal extract of the product. So as you can see from the picture there, the preclinical studies consist of, before we go for the clinical, so this is considers like a pipeline, for you before you go for clinical trials. So this is some part of the preclinical studies. Normally, this is how we develop we form our unit based on this preclinical pipeline. So starting with the with the traditional knowledge, followed by the phytochemistry analysis, characterization of the extract, followed by the efficacy, safety, before we go for the clinical trial. So in our center, we have the integrative medicine information unit. We also have the phytochemistry unit, where the function of this unit is to provide us the materials to be studied, for example, like the extract. And then the bioassay unit, the main function of the bioassay unit is to perform the efficacy studies to look into the pharmaceutical activities of plant, for example, like anti-cancer or the anti-inflammatory, antioxidant, something like that. And then once they gathered all the information to know that, okay, this plant have a potential to be developed further, with their pharmaceutical activities, then they will pass to our unit, toxicology and pharmacology unit. So our main role is to perform the in vitro and also in vivo toxicity studies. And then we also have this GLP section, good laboratory practice section, where it just formed 3-4 years ago. So the function of this section is to coordinate for the GLP studies. So let's say if you have any product with the therapeutic claim, as we know that in our regulatory, to register or to market your product with the therapeutic claims, you need to provide the safety data under the GLP certified lab. So we in IMR, we have the GLP certified lab. So if you have, or if any industry have this product to be developed to go for the clinical trials, so we offer the service to run the toxicity studies. Okay, so for today's presentation, normally I will start with the introduction about the toxicology, the definition, and then the history of the toxicology, why we need to perform the safety and toxicology studies, followed by the guidelines. So to perform the toxicology studies, we need the guidelines. So how to design the toxicology studies, what type of the toxicology studies needed, because sometimes with the different products, we need the different study design. You need the different guidelines. So let's say if you have a product for the orally intake, you plan for your product for oral intake. So the study design could be different with the intramuscular administration or intradermal administration. So it will be totally different. Let's say if you have a product for the topical use, so the guideline, the study design also could be different. Okay, then after that, I will share with you the basic requirements. Okay, let's say if you want to conduct the preclinical studies, so what requirements needed in terms of the duration, how long you want to conduct your studies, and then the type of animals, and then the route of administration, as well as what the parameters need to be measured in order for you to conclude whether your product is toxic or not, or maybe have a potential to produce the toxicity or not. Yeah, and then as I said before, I will share with all of you. Actually, some of the pictures in my presentation is based on my studies. So we have done the toxicology study for the in vivo, particularly on the prenatal developmental tox. So it's based on my project on the misai kucing, also the Ficus deltolia. Okay. Okay, so as I showed just now from the previous slide, so this is a normal, the standard herbal drug development plan, where normally it will start with the discovery. So let's say if you have the idea, okay, let's say when you look at the compendium, for example, you know that the traditional use of certain plant can use for example, for anti-cancer, like something like that. So from there, you start with the discovery first. You explore, see the potential, do with the in vitro studies first before you go for the total preclinical studies. With the complete preclinical studies, we include the efficacy and also for the safety. So normally, you can start with the efficacy study first. See the activities using whether the in vitro or the in vivo studies to see whether your this plant really have these pharmaceutical activities. Then once you gathered all the information, then you have to perform the safety, because there's no point sometimes at the dose that you selected for your efficacy study, but when you test it in the safety study, this particular concentration may produce the toxicity activity. So we don't want that. So that's why that's very important for you to run both study, the efficacy and the safety. And in fact, in the safety also, you can plan with the dosage. Let's say for your efficacy study, 2000 milligram per kilogram produce the efficacy. So in your safety study, you can play around with your dosing, the dose, So to make sure that, from there we know whether what particular concentration may have a potential to produce the toxicity activities. And then after that, once you get all this data, so basically the preclinical data is very necessary to develop this, they call it investigational brochure, where all the information about your product, about the formulation, about the standardization, about the efficacy data, about the safety data is required before you go for the clinical trials. Normally, like in the clinical trial, it consists of the phase 1, phase 2, and phase 3 study, which I'm not going to into detail for that. Before you can go for register, and then you can produce your product in mass production before you can sell, before you can let the consumer use your product with the therapeutic claims. Yeah. Okay, the preclinical studies. Preclinical studies can be defined, the reason why we run the preclinical studies is to determine the ultimate efficacy and safety profile of product before proceeding with the clinical trials, like I mentioned just now. So this is preclinical studies is a must. It's a very important. You need to provide the study from the preclinical studies before you proceed with the clinical trials, because from the preclinical studies, the data you receive, you can predict any possibilities could be happen when you run the safe clinical trials. Okay, and this include the characterization of the toxic effect to target organs. So the main reason we run the preclinical, the toxicity studies, is to see whether our product may have a potential to cause the toxicity in any target organ, for example, like a liver, our kidney, lungs, so many other vital organ in our body. So that's the main reasons why. And then another thing to see the dose dependence, and also to see the relationship, the exposure, whether the long exposure or the short exposure may have a similar potential toxicity effects or not. Okay. Okay, the toxicology can be defined, the study of the adverse effects of the chemical, which is like in herbal plant. We know that in herbal medicine, herbal plant, it consists of a lot of chemicals, compounds that can be toxic. A physical or the biological agent, for example, like a vaccine, on the living organism and the ecosystem. And the toxicity can be defined as the adverse end product of series of events that initiated by exposure. So by exposure, then we can see whether your product may have a potential to cause the toxicity. So that's the reason we have to perform the toxicology studies. Okay, the reasons why the toxicity safety studies is very important because of the history. So based on the history in the 1920s and early 30s, this is where the environmental mammalian territory started, where in 1941, one of the researchers discovered that the infection with the rubella virus during pregnancy could lead to the syndrome of congenital defects to the babies, as you can see from the pictures here. And then this is a big, it's not big issues, this is a big news on the thalidomide. And maybe I'm sure that maybe you know about this in early 1950s and 60s, where the effect of the drug thalidomide. So before this, in that particular years, pregnant women use this to treat the morning sickness. So they take this thalidomide to treat the morning sickness, and then surprisingly, this drug caused the limb reduction defects to the babies, for the legs and also for the legs and for the hands as well. So because what they believe, when they conducted the studies using the animals, so the animal looked fine, everything's look okay. But when you go for after the consumed by the human, so this is what happened. So this is the reasons why all the drugs or the herbal products or anything that to be consumed by the humans, especially to the pregnant woman. So this is why you need to conduct the preclinical studies, the safety studies, to make sure this is not happen again. So that's I think that's another reason why the toxicology studies is necessary for in the drug development. Okay, so when we talk about the herbal plant, so I'm sure that all of us think that, you know, when herbal plant is naturally formed, so it could be okay and could be safe. Yeah, so we have to remember that some of these medicinal plant used in herbal medicine contain chemicals or compound which can be potentially toxic. So a few studies have been conducted, and this is from my reviews in journals. So like one example is a blue cohort. So they found out that this blue cohort caused an infant, yeah, the report, an infant born with the acute myocardial infarction, profound congestive heart failure, and shock as a result of the mother ingesting blue cohort. Okay, and then another plant, sassafras, the oil of this plant is toxic in doses as small as 5 ml to adult humans. And then for the Chinese ginseng, they found out that the compound called GRB1 has the teratogenic effects during the mouse organogenic period. So this is where during the organogenesis, the development of the organs, when they use the whole rat embryo culture. And then last but not least, ginger. As you know that maybe ginger is okay, but a study in utero exposure to ginger tea results in increased early embryo loss with the increased growth in the surviving fetuses. So when they do the in vivo studies, in the animal study, they found out that a lot of the animals, the fetuses lost. Okay, so it caused the disturb the development of the embryo. Okay, so why toxicity studies are required in herbal drug development? Okay, the reasons, like I mentioned just now, is to provide the sufficient safety information to support the planned clinical use of the herbal products. So the first reasons why is to establish a safe and the immunogenic dose, to identify the potential toxicities and target organs, and to identify the safety parameters for the clinical monitoring. And number four is to evaluate the effect on the fertility and the fetal development. So this is all I will uh, present after this about the what type of the studies need to be conducted to answer all these requirements. Okay. Okay, so what studies are required for the preclinical safety studies to support the development herbal-based product? So for today, I'm going to talk about, to share with you what the common toxicology studies, especially when you're taking orally. If you have a product for orally taking, for oral administration, so this is a common toxicology studies need to be done. Case number one is the single acute toxicity studies, where basically this is only single administration. So you have the animals, you test it, you give one single dose, and then you observe the animal, what happened to the animals. Okay, number two is the repeated dose toxicity studies. So repeated dose means that every day you have to treat the animal with the same dose. In a repeated dose, they have different the 28 days, 90 days, and 120 days studies with the subacute, subchronic, and the chronic studies. So again, it depends on your product. Let's say if your product's required for the long term or the lifetime usage, so you need to perform your toxicity studies up to the chronic studies. Let's say if you product only for a few days, for example, like for 3 days or for 4 days, you don't have to perform your studies up to 90 to 1 year's studies. So only for acute and also for the 28 studies. So this is what you should know. The studies dependent on your product to be produced. Yeah. Number two is the reproductive and developmental toxicity studies. So for the GLP, this is one of the requirements as well, to perform the reproductive and developmental toxicity studies to see what happened to the pregnant mother, and also to see what happened to the fetus development. Number four, genome toxicity and carcinogenicity studies. And then number four is the safety pharmacology studies. So let's say, as I mentioned just now, let's say if you have a product with the different administration, like a topical administration. So you have a different kind of the toxicology studies, for example, skin irritation studies, corrosive studies. So you can use for the in vivo and the in vitro studies for this topical use. So it's slightly quite different with the normal oral administration studies. Okay, how to perform the studies? So I'm sure that we cannot just simply like, okay, I want to do the tox study and then I just do and take only 2 animals and test it. No, you have to follow the guidelines. So if you're not sure how to study your design for your tox study, so you can go to this guideline from the OECD guideline, Organization for the Economic Cooperation and Development, or OECD guidelines. So normally, like we are in our unit here, even the JLP, we normally we follow these OECD guidelines. So they have all different kind of guideline for the acute, for the subacute, for the in vitro, for the genotoxicity, for all. You can just Google OECD guideline. From there, you can see so many different guidelines that you can use to guide you to design your studies. And also from the FDA, Food and Drug Authorities, the FDA also provide the guidelines, WHO as well, and also the ICH guideline for the pharmaceuticals. So this is the 4 guidelines that can be used. Even though for the JLP, you can use any of these guidelines. So all the data produced, basically all these guidelines is quite similar. Maybe they have like something, but all they accepted the data if you follow this guideline. Yeah. Okay, so in toxicology studies, so they have 2 types, in vivo and in vitro. So normally we start with the in vitro studies first. This is like a screening to see whether the plant have a potential toxicity effects or not. So nowadays these zebrafish models has become popular to test for the screening, even for the efficacy studies as well. So, and then for the in vivo, I'm sure that you know that in vivo is the best model using uh, beagle dogs, for example, monkeys. And in fact, in the guideline, they also stated like, for the toxicology studies, for the repeated dose especially, so they are required for you to perform in 2 different animals, yeah, rodents and non-rodent. So so you have to bear in mind, like in a subchronic study, for the long period of study, the repeated dose. So in Malaysia, I think we only capable of doing for this GRP only for up to the rodents. So we do not perform any tox study for the non-rodent, especially beagle dogs or monkeys yet. So so that's why from based on my experience, a lot of the industry, the company, they do this the higher animals, non-rodent animals in India and in other countries with the GLP certified labs. So what here in Malaysia, we can do up to the rodent animals. Yeah. Okay. So for the basic clinical toxicology assessment, so for the study design should be adequate to identify and to characterize the potential toxic effect of the herbal product. So you have to know about the animal species, what type of animal species to use, whether you need to use mice or whether you need to use rats. Okay, for this rats, what the different types of rats. Okay, so like the different with the efficacy studies, sometimes even though in the efficacy studies they have a knockout rats or the animals, so focusing on this the disease. Let's say you want to treat your your herbal plant may have a anti-diabetic, so they can use the knockout animals with the anti-diabetic properties. So it's easy. Okay, so like toxicology, it is straightforward. So you can use just like a spot dolly rats, like a Wister rats, or mice to perform your studies. And then sex. Okay, so sometimes with the different studies, like an acute study, they only require 1 sex, okay, male or female. But for the repeated dose, they require for you to perform both, for males and females, right? Okay, and age as well. So it depends. They normally in the guideline they give the stated, the age, the range of the age, and also the range of the body weight. You cannot be too small, you cannot be too old, your animal, to run the safety studies. And then the size of groups. Okay, so like I mentioned just now, in the guideline, they stated, okay, at least you have 4 groups in a repeated dose. We have a control group, you need to have a lower dose group, medium group, and also the highest dose group. Okay, so this is, you have to know the requirements. So let's say if you didn't follow the guidelines, so let's say if you want to come up with the data, so you want to register products, so when they look at your data, it's not enough, you didn't follow the guideline. So maybe it can be difficult for you to register and also to commercialize your product. So that can be the main issue. Okay, and then also the dose. Okay, so in each guidelines, normally they will give you the suggestion what dose that you can be used. Okay, let's say if in your preliminary studies you have tested and then you said, okay, this particular dose may have the efficacy effect. So that particular dose you use for the toxicology. Let's say if you have no idea about your dose, you don't know whether what dose should be used with the lower dose, then in the guideline already stated, okay, the highest dose you should be used this particular dose. So let's say in that highest dose it didn't produce any toxicity effect, then you can consider that your product didn't cause any toxicity activities. Yeah. Okay, the route administration again. Okay, so if let's say if your product for the oral administration, there's no point you to use run the study for the intradermal. So you cannot use. Let's say you have a data on the intradermal, you cannot use. Okay, let's say you conducted your study intradermal before, and you say, okay, I just want to use this data to register my product with the oral intake. No, you cannot do that. So you have to run again your studies using that route of administration. Okay. So in fact, in the guideline also, they stated what the parameters need to be measured. For the biochemistry, you have to do the blood collection. So there's no point after you're treating your animal for a long period of time, and then during the necropsy, you said, well, I don't want to take the blood. I don't think so. It's really important to take the blood for the biochemistry. No, you have to take the biochemistry, and for the organ, you have to collect the organs, and then you have to take blood for the biochemistry and hematology. So from there, these parameters actually can indicate any changes to the parameter for the hematology and for biochemistry. You can see effects of your product for your plant extract into the animals. So, and then further you confirm, let's say there's some of the biochemistry related to the liver injury, for example. So from there, you do the histopathology, and then you can see whether it's really true or not, from your biochemistry activity, that the effect, it's related to the pathology on the liver, whether they can see any damage or not. So this is how you can make a conclusion after that, whether your products or extract is toxic or not. Okay, so for the single acute toxicity, the objective of the single acute to provide the preliminary tolerability, the safety data, and useful information in order to evaluate the acute or single exposure. So design for the single acute toxicity like this. It's only single administration, and then you observe for 14 days, and then you sacrifice the animal on the 15 days. So they've got a few guidelines on the acute toxicity on the OECD guideline. They call OECD guideline 420 fixed dose method, 4 to 3, and 4 to 5. So you can choose any guidelines. It's all accepted. Yeah. Okay. So like what we normally use in our JLP lab, in our unit as well. So we normally use this guideline 420. So it's simple. Basically, it started with the sighting study. So starting study started with the 5 milligram per kilogram. So we single dose, and then we observe for 24 hours. Let's say the animal still survive. Then we choose another animal, you treat with the 50 milligram per kilogram. So we observe 24 hours, if still okay, and then we increase up to the 2000. So let's say the 2000 we tested is okay, then we go for the main study, we include additional animal for using that particular dose. So that's last administration, then we observe the animal for 14 days. So during that period, we observe for any clinical behavior. So any changes to the behavior, whether um, the extract caused the diarrhea to the animals, or maybe the locomotor activities of the animals change. So the skin, the eyes. So this is all the parameter that we measured. Yeah. And then on day 15, we do the necropsy. So in the acute tox, normally they not required you to collect all the organs. Even they do not require you to collect the blood, because this acute study just to give you the ideas whether at what concentration your extract or herbal may produce the toxicity activities. Okay, for the repeated dose toxicity studies, so this is for the repeated dose. The objective is to observe, to find the non-observed adverse effect level, NOEL, which can be used for the establishing the safety. And as I mentioned, they have a short-term and also long-term, subacute, chronic, and subchronic studies. Okay, this is example of the 28 day studies. So you have to dose your animal for 28 days, 29 you necropsy, and then in fact this study, for the 28 day study, they have a satellite group. This satellite group just to see whether after you stop the administration of your product, whether it's reversibility or not, it reverse or not. Because sometimes, after 20, let's say for 28 days, it show the toxicity activity. But when you stop the administration, to leave it the animals for 14 days, the toxicity will revert back. So that means that there's no toxicity happen to the animal. So this is the design for the repeated dose 28 days studies. Yeah. So for the 90 days, it's longer, it's more challenging. The total numbers of animals, it can be around 100 animals. So you imagine when you want to do the necropsy for these animals, so you have to strategize. So you have to design properly how to perform the subchronic. So we haven't done any chronic studies. So as you know, that the chronic study can be even longer, because it take the animal involved, it will be around 300 animals. So you require a lot of manpowers to perform the chronic studies. Okay, next for the reproductive toxicology. So this is my research interest more on the reproductive toxicology. So this study, the toxicology, the reproductive, is to study the adverse effect of the substance or herbal plant on any aspect of the reproductive cycles. So we want to see whether your plant may produce the function of the reproductive system, or maybe your plant can disturb the development of the embryo, or maybe it also affect the development of the male or female reproductive activities. Okay, so they have a few guideline from the OECD guideline to study the reproductive toxicology. For example, the guideline 414 for the prenatal developmental toxicity. Then I will share with you after this. And then they have a 1 generation reproduction, and also they have a 2 generation. So that means that they expose the mother, the father, and then the mating, and then they produce the baby, they let the baby grows, and then after that they treat the baby as well, and see the generation of the babies, what happened if you're taking this extract. So whether they will change the genetic of the animals and will affect the reproductive activities. Okay, so for the prenatal developmental toxicities, to provide the general information concerning the effects of the prenatal exposure on the pregnant animals and on developing organism, which is the petals. So from the OECD guidelines, so they required 4 to 5 different groups, which one group consists of the 20 pregnant rat. So normally we started with the mating, so with the 2 female and with 1 male. So you mate, and then the next morning, overnight, normally you're mating overnight, and then the next morning you have to do the vaginal smear to see whether the presence of the sperm in the vaginal smear. So let's say when you look at the, under the microscope, they got sperm on the slide, on the vaginal smear, we can consider as a day 0 of pregnancy. So normally some different study, they do the administration on the JD 0 until 20. And some they start to administer the herbal extract on the JD6, the pregnancy day 6 up to the 20. So because the period of time during JD6 up to 20, this is where the development of the organs. Okay, so we want to see the effect of the extract in the whole process during the organogenesis So we treat administer the plant on this period. And then on GD 21, so we have to sacrifice the animal, do the C-section. If let's say if you miss this 21, because sometimes for this rat, normally on day 21, day 22, if you do not perform the necropsy, the cesarean section, so they will deliver naturally. So that one you have to exclude it, because you need to do the necropsy, because you have to see the placenta. You have to weigh the placenta. You have to see the animals, and then in fact the placenta. Let's see if they have any resorption to the fetuses which is not developed well, so you can see from the gravid uterus. So this is the reason why you have to see the section, and then you count the animals, and then you see the sexes, the gender, how many male and females rat. Then from there, and then also you have to do the morphological examination to see the hands, the head, the shape of the head, the nose, something like that. So to see the effect of your plant. Okay, so this is some of the example of the parameters need to be measured. So the maternal reproductive performance and also the fetal parameters. We want to see both effect of the extract into the pregnant mother, what happened, and then also the fetal parameters. So you want to see the body weight, the gravid uterus, and then also the food intakes and so on. Yeah. So this is how we perform the budget smear. As you know that rats consist of, they also have an estrus cycle. So they only 4 cycles. So they started with the estrus, met-estrus, di-estrus, and pro-estrus. So normally during the pro-estrus is a good time for you to do the mating with the male animals. So they're more receptive to the male. So let's say if they're not in the right cycle, so you introduce to the male, say, as you know that this female rat will kick out the male, and then we get fight with the male rats because they're not ready to receive with the male rat. So normally like we do, because if you want to see every day, you have to do the vaginal smear and see at what cycle your rat is before you introduce to the male rat. So normally during the pro-estrus, you introduce to this, as you can see from the picture here. Okay, so when you introduce, so the male will explore the genital. They will, trying to see, what they can do, and then they will start the mounting, and then also it will have an ejaculation. Okay, so this is not happen one try. So normally they will try again and again before this ejaculation can happen. So this is, and then the next day we will do the vaginal smear and see the presence of the sperm. Okay, so this is administration. Normally we do the orally to the rat, the pregnant rat. So the necropsy we perform on the day 21. So this is how it looks like. So this is a normal pregnancy. This is the gravid uterus, and then you open the gravid uterus, and then you count the number of the fetuses. Sometimes you can find more than 10 fetuses, and then you have to weigh the placenta, and then also the fetuses, and then you see any abnormalities to the fetuses. Okay, so this is the fetal external follicle. So this is the example of the malformation happens. So this is the late resorption. Normally, the stop development during the pregnancy. So you can see the development is like this, and then sometimes the fetuses didn't grow well. So it's slightly smaller than the normal fetuses. And then this is how the method, how you differentiate between a female and a male fetus based on the anogenital distance. Because sometimes, let's say if your product, if your extract have the phytoestrogenic activities, so even though in a female rat also may produce a longer anogenital distance. So because normally we don't want that the plant have a phytoestrogen, because it will change the characteristic of the female and also the male. Yeah. Okay, so after that, half of the skeletal, the fetuses, we go for the skeletal examination. So normally we stain the whole fetuses, and then we look at any abnormalities on the bone development. Okay, so we use the special alizarin rat, the stain, and then from there you examine. So this is all the this is the normal normal head, and this is some of the abnormal. So you can see that the absorption of the stain, it's related to the bone development. Okay, so this is the frontal bone, parietals, and the hypody. So you imagine how many fetuses you have to collect, and you have to stain, and you have to examine for each of the part of the fetuses. And this is the example of the ribs. So this is normal ribs. As you can see, this is like a wavy ribs from the fetals, and also vertebral column. So sometimes they have a split bones here, and then the forelimbs, the resorption of the stain is lesser than the control. And sometimes it's not surprising. Sometimes, even though in the control animal, fetus also may produce this, and from there we can decide based on the second analysis whether your product is toxic on the embryo development or not. Okay, this is another half. We cut with the chemicals, and then we cut the head of the fetuses, and we look any development, abnormal congenital development. For example, like here, we found one cleft palate. So if you didn't cut this, we don't know whether your extract may produce this or not. So this is likely we found only one fetus with this cleft palate. Yeah. Okay, for the genotoxicity studies, so the study of the adverse effect of the genetic materials, DNA caused by genotoxic substance which may affect the integrity of the cells. So that's the reason why we perform this genotoxicity effect. This is also the must. This is the battery testing. Normally in the regulatory, even internationally, so they require you to provide this result from at least 2 in vitro study and 1 in vivo study for the genotox, for you to register your products. So this is a common genotox study that people normally use to see the genotoxic potential. So they're using this bacterial reversion test, AIMS test. So basically they use a different strains of the salmonella, the mutant salmonella, and then see whether any is reversibility, whether they can revert back to the normal salmonella. And then for the in vitro and in vivo micronucleus assay, this is we expose together with the positive control. We want to see any micronucleus found in the bone marrow. Okay, after the certain period of the time, so we check the bone marrow. We start take the bone marrow, and then we measure the numbers of the micronucleus. Okay, for this is another common assay for the genotoxicity activities. Okay, safety pharmacology studies, it depends to evaluate the potential for the undesirable secondary action that affect the vital function such as central nervous systems, respiratory, cardiovascular, and renal function. So let's say if you perform the previous study, the general toxicology, and if you find that your extract may produce this on this particular system, respiratory or cardiovascular or renal function, then the safety pharmacology maybe is required to get the data of the safety. Okay, just before I end my presentation, so just like it's not like a promotion, just I want to share with you that we have, this is our non-clinical research facility with the JLP certified lab. So this is situated in Jalan Pahang in our old building. So we do have this GLP certified lab. So this is our normal activity where we have this hematology with the GLP facility with the biochemistry, and then we do have experimental rooms. If you want to perform the animal study, we can provide that. And then this is the feeding, and also this is a histopathology, and also the archivist. So normally for so far our sample, for the histology samples, we hired the histopathologist, the expert to read the slides. So but we prepare all the slides for ourselves. So we do have the facilities. Let's say if you have products, or you know that somebody in the industry wanted to perform the safety with the GLP study, then you can come to us and we can discuss further. Okay, another thing that we also just established this post-implantation of rat embryo, whole embryo culture. So this is the first in Malaysia. So one of our officers managed to establish this technique where normally they um, mating the animals, the male and female rats, and then on day 9 of pregnancy, so they have to do the necropsy, so they collect the implantation, the embryo, and then they growth the embryo outside using the system, and then we expose the extract at different concentration. From there, you monitor the development of the embryo, whether you can see the toxicity activity of your extract of the plant. Okay, we also last year we also managed to establish this zebrafish laboratory. Now we are working on the toxicity activity using zebrafish models, and also we also just started working on the efficacy study as well on the neuroprotective activities using the herbal plant. So again, like I'm open for any collaboration. If you're interested to come to visit and to work with us, not only with my unit or with the other unit as well, or other centers in IMR, you are most welcome to come here to see us. With that, thank you very much for your attention. So I got the email address here and the phone number, so you can contact me at any time. Thank you.
Thank you, Dr. Hussein, for a very informative and interesting talk. And also thank you for our audience for this support our webinar. And then, yeah, it's very interesting. We conduct with clinical and clinical, and we hope that we can join or collaborate something with IMR in future. So, Dr. Hussein, I have a question for you before I read through from our Facebook. And just like you mentioned just now, for example, if in herbal supplement, we know that the environment is being controlled. Can be, I mean, is there any chance that we can be toxic in a way?
Yes. So basically, like you said, even though the production of the herbal can is control environment, sometimes you have to remember that let's say you produce the extract, so the contamination could be happen, and then in fact, like even the environmental also, like from the soil, the heavy metals, the contamination for the microbe as well. So in fact, the reasons why normally before you produce your product, the stability testing is required, to see whether your product is stable or not, because sometimes the temperature may affect the production of the microbe, the aflatoxin, for example. So this is things like you have to take into consideration to make sure that your product is really free from this, which may produce the toxicity effect when you consume the product.
Yeah,
So
I agree with that. Yeah, and
yeah.
while waiting for our audience to know some of the question, so oh yeah, we have one question for Dr. Hussein. Okay, from Farah Aman. Salam, Doctor. What is your opinion on the toxicity and efficacy of multi herbs utilization in one product, which is either topical or oral? So,
Okay.
what's your opinion, Doctor?
Thank you, Farah. So for this one, we do have like some of the researchers that I know developing this product using the multi herbs. So normally for the multi herbs, the requirement whether you have to do the toxicity study for the whole extract. Let's say in one capsule, they might have a different plant inside the capsule. So you have to test that final formulation to do the toxicity study. So basically, it's the same. Okay, to the study design and the efficacy study could be similar with the single herbs. So yeah, that's my opinion on that. So because normally people, they put more herbs, because let's say one herb may produce this effect, second herb this effect. So they want to have a combination to make it that's really effective. So they do the combination of the herbs. They call the multi herbs products. So yeah, you still need to do same toxicity and efficacy studies. Yeah.
Okay. Is there any question? Yeah, we have one from Mr. Amirul. Good morning, Doctor Hussein. Yeah, what are the symptoms of herbal overdose?
Okay,
Normally, what
morning,
symptom?
Amirul. So normally for the animals, so based on my experience in animals, when let's see if they have a potential with the highest dose, the toxicity what you can see is the locomotor activity, and sometimes the diarrhea. The animal can have a diarrhea, non-stop diarrhea, and sometimes they may reduce the body weight, and in fact the water and the food intake also can be reduced. So that's another symptoms in the animal. So I think it's similar to the human. Let's say if you overdose sometimes, any products, you may produce the diarrhea, you not feel good, you see. So this could be the symptom, or maybe headaches or fever. This could be the symptoms of the herbal, the overdose.
Dr. Hussein, if we have that symptom, do you mean that herbal is not good for you, or the herbal supplement is not good for you, or the product is contaminated already?
Because sometimes you have to know that, because not all herbs or even drugs, so that's why, with the different people, we cannot like one herbs. They say, you, Dr. Zuraini, you may have a similar effect with me, maybe for you okay, with me no, I'm not okay. So that's why sometimes, this, to make sure that's not overdose or produce any toxicity effect, you have to, the dosing. So that's why it's based on your body weight. So that's why, we
Toxicity is
cannot
very
say like
important.
whether it's good or not, but it depends on the individual.
That's why we encourage people to have knowledge on toxicity study as well.
Yes, yes. You cannot just simply buy the product, especially because now I think the people so good. They said, okay, they got the idea. Okay, this is traditional use for this. Why not we come up with the product with the supplement? Because they know that if you want to have a therapeutic claim, the process could be, the scientific study you need to perform is longer. So
Yeah.
just put the supplement, and then the data for the supplement products. It's only for heavy metals and microbial content. If this is free, then you can register a product with the supplement claim. So, but again, people, even though the supplement, people will say like, oh, this is good for the anti-cancer, for the diabetic, even though they have not proven for that, the scientific evidence. So that's why I think we need to educate our people, to understand about this. Yeah.
Okay. Is there any question? Ada question lagi? Yeah. Okay. One more question from Sureni Shahid. Okay. Salam doctor. What are the source of toxicity in herbal medicine? Source of toxicity.
Okay,
Sources.
thank you, Surani. So, the sort of the toxicity can come from the environment, and also can come from the plant itself. So, certain plant, they have the toxic compounds in the plant, which may can cause the toxicity effect. So that's why sometimes, in research, with the advanced technology, so they manage to extract out the toxic compound. For example, I'll give you an example like obigadon. So we know that obigadon can be toxic. So that's why traditionally used, people normally, the obigadon, they will put in the running water for 1 week to extract the toxic compound. So let's say now with the technology, we can extract out the toxic compound and can be used the rest of the obigadon, which we know that can have a different anti efficacy activities. So yeah, that can be come from the herbal medicine itself, the herbal plant itself, and also can come from the contamination, like I said just now, the contamination from the bacterial, from the aflatoxin, or from maybe from the during the processing of the preparation of the herbal medicine.
All right. Dr. Hussein, with the advanced technology, can we see that AI, okay, for AI, is there any chance that in the future that animals, sectioning is not relevant anymore, or do you still thinking that we still need this animal to conduct the toxicity study?
My personal, because like last year we heard about in US, they passed about the planning to stop the use of the animal. But personally, I think we still need to use the animal model, because of what, it's different. Even though in vitro studies, we can just predict, but because like the animal uh, can slightly represent, the human. So for the prediction of the toxicity, a study can be found from the animal model, because now a lot of people, they are now going into like the use of zebrafish, and then use of organ in chips. They put like a chips, and then let's say they want a kidney, then they can create the chips like a kidney, and then they can expose. From there, they can collect the
Mimic. Ah,
sorry,
they mimic
anything. Yeah,
they
the
mimic.
mimic. Even the kidney, and then the lung, so liver. So they then put in chips, and then exactly the mechanism could be similar, but how can I say? Whether we can accept that now, I don't know. But in future, we don't know with the advanced technology. So maybe
The
all
policy, yeah.
maybe this kind of technology can replace the use of the animals. So,
Yeah,
but for
Doctor.
now, we still need.
Yeah, Doctor. Okay, thank you for that. We have one more question from Lina. Okay, good morning, Dr. Hussein. Thank you so much for sharing. How do plant develop tolerance to toxicity? Thank you.
How to plant develop tolerance.
Tolerance. Mm.
So this is how to. I'm not really sure about these questions.
Mhm.
Tolerance to the plants or to us. If let's say for the plant tolerant to the toxicities, because normally you know that plant also have the defensive mechanism, so they have their own compound that can be use the antioxidant. So that's why this is the antioxidant with all this compound we use for us as well for the for our defense. Let's say like a product plant for the anti-cancer, for example. So this is one that maybe the compound produced from the plant can be benefit to us as well for with the reach of the antioxidant activities. So this is what I can,
Fair enough. Yeah.
if I
Okay.
answer.
Yeah. If we have any more question, any question? Huh? Okay, we have one more. The Hussein. Oh, yeah, you have so many questions here. So we would like to ask from Mida Madiha, Salam Dr. Hussein. Okay, does it necessary to comply with all the guidelines for toxicity, or we can choose either one? Thank you so much.
Okay, thank you, Ida Madiha. So as I said just now, let's say if you have a product for with the therapeutic claims, you want to register your product to commercialize with the therapeutic claim, you have to do the toxicology study. Only toxicology need the JLP, but the efficacy study is okay. So the JLP, the tox study for the safety, you have to do the JLP studies with that guideline that I mentioned just now. So you don't have to do it all. The guideline is depend, as I said, depends on your product. So let's say your product for the short duration usage. So you don't have to go to all guidelines. Only look for the acute and also for the subchronic for the 28 days. You don't have to perform your tox study up to the 90 or 1 year study. No, it's not required. So only depend based on your products, what your plan for the clinical trial. Yes.
Okay, thank you. We can have like idea how much is that cost for that for one single
Okay.
dose.
Okay, for the JLP, normally it costs a lot, 100,000. So
Mm.
even we have up to subchronic study, acute, subacute, and subchronic cost like almost like 900,000 ringgit because
Wow.
animal normally like we are here in MR, so we bought the SPF, the specific pathogen free animal. So we bought it from the GLP certified lab who produced the
Mhm.
animal. So sometimes the cost of the traveling, aeroplane, so you have to think about that all the cost. So that's why it can be a bit expensive. Yeah.
Oh, Dr. Hussein, we have one more question from Profida. Assalamualaikum, doctor. Let's say your end product got contaminated with trace or compound which may be toxic. However, you didn't use it in any of the raw materials or mixture. So, and from your analysis, none of them got that puzzling compound. How can you investigate the source of contamination to avoid in future?
Let
Yeah,
me read
we have
again.
like, yeah. Can you read through from our...
Never got contaminated with trace of compound.
Mm.
However, you didn't use in any of the raw material mixture. None of them got. So basically, what Ida used asked me, the end product got contaminated with the trace of compound, which may be toxic. However, you didn't use any raw material.
Oh, I mean, it traces in our product, which is we not having that in our ingredient or any of one of it in our compound. So, how to investigate the source?
So that's why normally what we do, you for the product we have to go for the antimicrobial test and also the heavy metal testing. So normally for the product we send to the lab to check for the any contamination of them, because sometimes you see sometimes your material is okay, after certain of period of time after you prepare the whole extract for the product. So this is might happen. You see the contamination from from the microbe, for example. So that's why normally what we do, we have to send the sample to the lab to check for any contamination for the microbial or for the heavy metals. Yeah.
Hope it helps. If not, we can still contact Dr. Hussein for further clarification for this question.
Sure.
So, and then we
Please.
have one more question from Dr. Hasmaliana. So, welcome, Doctor. Thank you for the nice sharing. I have a few questions. The first one is, in your opinion, how effective herbal product in comparison to prescribed drugs? Okay. And the second one, can we just consume herbal product instead of taking prescribed medicine? For example, high blood pressure, diabetic drugs, and so on. And the third one is, or is it okay to consume herbal product together with the prescribed medicine? So can we do that? So
Okay. Okay, for the first question, I have how effective herbal product in comparison in prescribed drugs. So my opinion, because there's no really concrete studies have been conducted with the comparison, with the prescribed drugs and also the herbal drugs. Okay, sometimes we only for the efficacy studies, normally we include the normal drugs as a positive control to compare with the herbal drugs. So sometimes, our finding shows that the herbal products sometimes can be comparable and sometimes a little bit lower than the prescribed drugs. I think this is because of normally the synthetic drugs, they have gone through all the studies, the research on that prescribed drug compared with the herbal drugs. I think for the herbal products, a lot of more studies need to be performed in order for us to to compare with the prescribed drugs, and also maybe can be introduced in the hospital. So a lot of the studies need to be done on that. And then can we consume herbal product instead of taking? So my personal opinion, we still need to, let's say it has a supplement, yes. Maybe you don't have to, you have to taking with the prescribed drugs. So let's say your hospital or this prescribe you with the blood pressure drugs, you have to still taking. Do not leave your prescribed medicine. So you have to still taking your prescribed medicine with the herbal supplement. Okay, is it okay to consume herbal product together with the this is again because like they have a studies about this warfarin drug. They have a interaction with the St. John's wort tea, the plant. See, you have to know. So that's why I think we have to know first before taking any medicine together with the herbal product. Sometimes they have a drug herbs interaction, which can give harm to you, because the warfarin, when you do the St. John's wort, that the warfarin, the function is what the blood.
The drugs, the blood, dilute the blood.
Yes, yes. So if you use the St. John's wort, it will get worse. So it can keep harmful to you. So that's why it depends. Some herbal okay to take with the prescribed medicine, but you have to know. So yeah,
Right.
that's my.
So any question from the floor, audience? Is there any? There's so many questions, Dr. Hussein. Okay, bear with us. We still have maybe one more question. Okay, one more question. So, okay, salam and good morning, doctor. Is it safe to consume the food containing herb that categorized under food, but not up to drug dosage? Thank you.
Okay, so we know that some food they may contain of a little bit of the herbs. You see, even now we also are taking like herbal all the herbs in as our food. I think I can say that it could be safe to consume, because during for the animal study, normally what we tested, even though up to like 500 milligram per kilogram, when we converted back to the human dose, it's too big. We're not going to take the herbal at that particular amount per day. So normally for the small amount could be considered okay. But again, like I said, just even from my slide of presentation, the cases where they take the oil of the sassafras, the oil also may produce, even a little bit of the oil may produce, give the harmful to when you're taking it. So again,
not too much.
not so much, but so far, when you're taking traditionally, it should be okay. Yeah, but so far we don't have heard any report on it. Yeah, so Yeah, so again, that's very important to research for all these particular plants to see when the potential of the toxicity activities. Yeah, so
Well, I think this is the end. Our question for Q&A for on our webinar. Okay, and also we would like to thank Dr. Hussein for a very informative and interesting talk about your study and about what IMR can benefit for us, and also and for our audience for support this webinar. And once again,
thank you so
we
much.
hope that you will bear with us in our webinar. And then thank you. I would like to invite our director to wrap up this webinar.
Assalamu alaikum, Dr. Hussein. Thank you very much for this very, very interesting. I think one of the highly questioned seminar we have in IBD with very high interest, updating our knowledge about the safety assessment for the herbal products according to the new guidelines in Malaysia. And I think you enrich our knowledge today a lot about better understanding.
Thank you.
And we are looking to welcome you here in IBD very soon, physically.
Inshallah.
And with this, I would like to say thank you very much again for your seminar today. Thank you.
Thank you very much, Prof.
Thank you.
Thank you.
Thank you. Assalamualaikum.
Inshallah.